Good morning, good afternoon, and good evening, everyone, and welcome to our webinar titled Sensorimotor Network Development During Early Postnatal Life in Awake and Sleeping Brain. This is Liam Sanyo from Inside Scientific, and I'm very pleased to be your host for today's event. This session has been sponsored by Neuratar, so a big thanks to them for making this possible. Today, we're very fortunate to have Dr. Anna Cavaccini and Dr. Jimmy Dooley with us, respectively, from Professor Karianis' lab at University of Zurich and assistant research scientist in the laboratory of Professor Mark Bloomberg at the University of Iowa. Today, Dr. Cavaccini and Dr. Dooley will share insights into the development of rodent sensorimotor neuronal circuits during early postnatal life in wakefulness and REM sleep. I'm really looking forward to getting started today, but first, just a few notes on housekeeping. So first, if the panels on your screen look too big or too small, you can zoom in or out in your internet browser to adjust their size. Also, the webinar is being recorded and all resources will be made available following the event. We'd love to hear from you, so please send us any questions, thoughts, or comments using the Ask a Question panel shaking on your screen there. We will be answering a few questions after the presentations. If you'd like to learn more about the research or the technology being presented today, we invite you to check out the Resources tab, where we've added a few links and downloads associated with the event. We do have a few polls and a post-event survey lined up, and we'd love to get your feedback. And finally, if you do happen to have a technical issue, refreshing the page is usually enough to get things working again, but if not, feel free to reach out to us using the Ask a Question panel, and we will help you to troubleshoot right away. And with that, I'm very pleased to welcome our first presenter, Dr. Anna Cavaccini. Anna, thanks so much for joining us today, and the floor is yours whenever you're ready.
Okay, thanks a lot. So, hi everyone. And as first, I would like to thank Neuratar for organizing and inviting me at that interesting webinar. Today, I'm going to illustrate you part of the project I'm currently working on at Zurich University in Professor Karianis' lab, and that is taking advantage by the use of the mobile on cage developed from Neuratar. More in detail, I'm working in a mouse model, and I'm focusing on striatum, which is a brain area important for the initiation and the execution of movement. A large piece of literature has evaluated the striatal function in motor control in adult mice. However, this topic has not been analyzed a lot from the developmental perspective. This talk will follow the structure reported, so I'll try to give you some background about the topic before moving to the central hypothesis and the aims. Finally, and furthermore, I will give you some information about the methods before describing the results and the conclusion. And finally, we'll go through the future perspective, describing what we are actually doing and planning to do. And this might be an important point of discussion. So let's start with the introduction. We interact with the environment through movement. Now the question is why it's important to have a clear idea of the development of movement initiation, execution, and termination. And the answer is that a proper movement development is going to impact the repertoire of action that we will perform during our life. And this is true in physiological condition, but this is also affected in neurodevelopmental disease as hyperactivity disorders, Tourette syndrome, obsessive compulsive disorders, and so on. A large body of studies has widely shown the role played by the striatum, the main input nucleus of the basal ganglia loop, in motor control. Basically, spinal cord circuits controlling muscles receive inputs from brainstem and midbrain nuclei and from cortex. Those inputs are tonically inhibited by the output of the basal ganglia loop, preventing the occurrence of involuntary movements. The activation of the striatum leads to movement performance and control different aspects of movement initiation, like selection, timing, or invigoration of upcoming movement. Now, this has been, as I said, well characterized in adult mice. I will refer to movement as in locomotion, And it has been shown that at postnatal day 10, locomotion is not fully developed. As you can see from the picture showing a postnatal day 10 mouse moving in an open field on the left, compared to a postnatal day 28 mouse moving in an open field as well on the right. And you can easily appreciate from the picture here that locomotion's trajectory in a postnatal day 28 mouse is much more complex. Now, this time window is important for the development of locomotion, but I found it very interesting because it also corresponds to the onset of different sensory modalities. Indeed, ears open at postnatal day 12, while eyes open around postnatal day 15, as well as the active whisking begins, which means that the whisker-dependent exploratory behavior appears. Striatum receive input from motor cortex. However, the cortical areas responding to sensory visual, auditory stimuli, as well as the somatosensory cortex S1 and S2 project to striatum 2. And all these excitatory projections make striatum an interesting subcortical hub for sensory and motor information integration. But this is true in adult mice. In terms of excitatory and inhibitory inputs to the striatum, something has been reported over development. Some studies reported the changes in the excitatory and inhibitory synapses on striatal projection neuron. They also pointed to the role played by glutamate in synaptogenesis at those circuits. Furthermore, changes in morphology and intrinsic properties of striatal neurons have been described as well. In a time window generally going from postnatal day 10 to postnatal day 21, so more or less the same time window I was talking about before. Now, with the hope that introduction was clear enough, I will move to the central hypothesis and the aim. If we only focus at cortical level, those areas are not independent station deputy to specific function, but there is an interplay between them that change over development. For example, it is known that in adult mice, there is an interplay between the primary somatosensory cortex S1 and the primary motor cortex M1, with sensory inputs arriving to S1 from the thalamus and then going from S1 to M1, thus modulating the motor output. However, motor maps are not detectable until postnatal day 25 in rats, and before, M1 seems to work as a sensory areas. And these changes in S1 and the M1 crosstalk have been also described by Bloomberg Labs, who's the next speaker Dr. Dooley is part of. Indeed, they have recently shown that while at postnatal day 8 rat sensory input can be processed following parallel pathways, by postnatal day 12, depending on the modality of sensory transmission, sensory inputs can be processed following parallel pathways or serial pathways. This means that the stimulus that lead to complex action selection that involves cortical processing in adult mice will be processed differentially by neonatal brain. And all this information suggests that early on, cortex is not fully developed. However, thalamocortical afferents relying information to sensory cortex are already established. I have already told you that at striatal level, it seems that early on, the number of excitatory synapses seems to be reduced. So the possible scenario early on might be that the thalamic inputs dominate over the cortical ones in driving striatal activity, thus modifying basal ganglia loop output and affecting the locomotion. So, as first, we will aim at evaluating the striatal output over development and evaluating the correlation of striatal output with locomotion over development. And this last point might be useful to have an idea about when motor maps start to appear in the striatum. Now, I will move to the methods. And as first, I need to spend some time on the methods because we want to record the neuronal activity in striatum in locomoting mice pups. However, there are some issues that are linked to this, complicating the achievement of this goal. So we would need to perform acute experiment in head-fixed mice. Why? Because mice pups cannot carry an implant, since it would be too heavy. And also, mothers cannibalize pups with implants, so the implant cannot be chronic. Furthermore, since we want to track locomotion, we need to use a system that mouse pups that are not too strong can easily move. And to do that, we took advantage by the use of the mobile-on cage developed from Neurotar. Indeed, mouse pups can easily move the airlifted platform. Furthermore, mobile-on cage is supported with a locomotion tracking system, providing directly some measurement as speed or angle rotation of the mouse and so on. Simultaneously, we performed silicon probe recordings of multi-unit activity in striatum. And here on the right, you can see the picture of the experimental setup we used. Now, let's go to the results. I showed you this slide before in the introduction, referring to the locomotion of mice freely moving in the open field. So as first, we tried to confirm those results, but in a different experimental settings, since, as I told you, mice here were head fixating and moving on an airlifted platform of the mobile-on cage, so they were not freely moving. We took into account 3 different time windows that are linked to the onset of different sensory modalities. So we have the first group between postnatal day 11, postnatal day 12, which correspond to before eyes opening. The second group goes from postnatal day 13 to postnatal day 15, and this corresponds to when the mice open their eyes and also when the active whisking occurs. And the third group goes from postnatal day 17 to postnatal day 24, and this corresponds to young adult mice. Already from the locomotion trajectories reported above, you can appreciate the locomotion changes at these 3 different time windows. And we measured the speed, and as you can see, the average speed calculated over 30 minutes recording progressively increased over the 3 time window considered. And these results were in line with what reported by Vanderburg in freely moving mice. We also measured the travel distance at these 3 time windows, and the average travel distance progressively increases over the 3 time windows considering, as it was for the speed. Furthermore, we calculated the cumulative travel distance over 30 minutes recording for each mouse. And here you can see this heat map where each row is a mouse. And I also indicated when the 3 time window started. And as you can see, the exploratory behavior seems to slightly increase between postnatal day 13, postnatal day 15, while it mostly increased at postnatal day 17, postnatal day 24. We also wanted to measure striatal activity, so we performed silicon probe recordings. We use a NeuroNexus probe composed by 8 shanks with 8 electrodes for each shank. The probe was impregnated with dye before the insertion in order to mark the insertion site. Basically, we inserted the probe at the level of the posterior striatum, which is rich in motor and sensory projection coming from the cortex. And after the probe insertion, we wait 15 minutes to allow the tissue to adapt to the probe, and then we record the activity for 1 hour. After the recording, the brain was collected and processed for immunohistochemistry to verify the right insertion of the probe, but also to select the shank that were inserted into the striatum. And first of all, we measured the average firing rate in hertz over 1 hour of recording at this 3 time window. And as you can see, the firing rate significantly increased at postnatal day 17-19 compared to the postnatal day 11, postnatal day 12. We also wanted to measure the correlation of the multi-unit activity time course with the speed time course, with the idea to extrapolate more information about the role played by the striatum in locomotion early on. And to do that, we did a cross correlation by using a function that allow us to check for similarities between two discrete signals by correlating one signal, in this case the multi-unit activity time course, with the lagged copies of the speed. And for each mouse, we selected the channels showing a correlation coefficient equal to the 90% of the max, where the lagged signals were restricted to more or less plus or minus 10 seconds. And so here there is the picture of an example of the two-time course. We measured the cross correlation, and what we found was that the striatal multi-unit activity show a higher correlation with the speed in young adult mice and a lower correlation early on. So what is the conclusion? Mouse pup locomote less than young adult mice, that's considering the speed, the travel distance, and also show a less exploratory behavior than young adult mice. Early on, striatal firing rate is decreased compared to young adult mice, and this might suggest a different engagement of the striatum from the excitatory inputs. And early on, the activity seems to be less correlated to locomotion. So it seems that striatal activity and locomotion are uncoupled, and striatal activity correlation with locomotion seems to go hand by hand with the cortical development. So what we are planning to do then? We are trying to better characterize anatomically the inputs engaging the striatum at the different time points, not only the cortical inputs, but also the thalamic inputs. But we are also trying to have a better idea of striatal activity and to understand how it relates to locomotion early on. So we are evaluating and comparing patterns of activity specific for the initiation, the execution, and termination of movement in young adult mice and in mouse pups. And we are also trying to go more into detail of striatal circuits and striatal cell through spike sorting of the silicon probe data. Furthermore, we are performing patch clamp recording on ex vivo acute brain slices with the aim to have a better idea also of the input recruiting, specifically the two striatal neuronal subpopulation. And with that, I would like to thank my supervisor, Professor Karayannis, for the exciting scientific discussion and for the supporting me in that project. And I also would like to thank all my wonderful colleagues, in particular George and Gwen, who are performing the analysis of the data, the anatomical data, and the characterization of the input engaging the striatum over development that I could not show you yet, but I hope to show you next time if I will have the opportunity. And last but not least, I would like to thank Neurotar again and all of you for your attention, with the hope that you enjoy the talks. Thanks a lot.
Fantastic. Well, thank you, Dr. Cavaccini, for a really fantastic presentation.
Thank you.
Before we move on to Dr. Jimmy Dooley, we do have two quick audience polls we'd like to ask. The first of which being, which of the following research methods do you currently use? So please select all that apply here, with the options being 2-photon imaging, electrophysiological recordings, behavioral assays, optogenetics, functional ultrasound imaging, wide field imaging, or click that last option if you don't currently use any of the above. And we'll just keep the options open for a few last moments to collect your answers here. Fantastic. All right. Next question And this is for those of you who currently work with awake rodents. Which of the following do you use? So please select all that apply here, options being full body restraint, head-mounted electrophysiological probes, other head-mounted imaging devices like the miniscope or Inscopics, airlifted ball or treadmill that's commercially available, airlifted ball or treadmill that's custom made, or pick that last option if it's not applicable and if you don't work in awake rodents. And again, we'll just keep these options open, or the polls open, for a few more moments to collect those answers. All right, fantastic. And with that, I'm very pleased to welcome the next presenter, Dr. Jimmy Dooley. Jimmy, thanks so much for being with us today. And the floor is yours whenever you're ready.
Yeah, thank you very much for having me. And it's great to be part of a session where there's more people studying sensory motor development. I'm Jimmy Dooley. I am an assistant research scientist in Mark Blumberg's lab at the University of Iowa, and I'm here to tell you about the developmental emergence of cerebellar models of movement revealed during active sleep. So I want to open up by just showing you a video of my dog Zelda. And this is every single day, right before I feed her, I give her 2 treats that also function as vitamins. And she's very motivated to eat these treats as quickly and as efficiently as possible, because that's how she can get to her food bowl. And so I'm just going to show you, to mix it up, often I'll throw the treat near her head, but not exactly at her, and see if she can catch it. So let's just watch slow motion footage of Zelda catching one of her treats. So here you can see that her tail's wagging. She sees that I have it, and she reaches, opens her mouth, and catches the treat. So I want to show you now her catching the second treat that day, but this time I want you to pay attention to how exactly she moves her head, and how she moves it not to where the treat is, but where the treat's going to be. And she anticipates the movement of the treat and kind of guides her head to where it'll intercept with that treat's path. So here's Zelda again, still happy, still paying close attention to me. And she moves her head to the side and opens her mouth and just lets the treat go into her mouth as it's moving. This is something that all of us with pets have seen them do, and it's something that, we might take for granted, but when you think about it, it's actually pretty complicated, because you have to be able to anticipate the treat and anticipate your own motor commands to meet the treat in the air. So now I'm going to show you another example, and this is going to be a toddler grabbing a ball. And so the blue ball there, this parent's going to put it on this circular path, and the toddler's going to be trying to grab it. And let's see how this toddler does. So here's the ball. It's put on this path, and he misses it once, twice, 3 times, 4, and 1 final time. So every single time the toddler reaches for this ball, he's always a bit behind it. And that's why this task is challenging, because every single time he moves his hand, it's where the ball was, not where the ball had moved by the time his hand got there. And this highlights one of the biggest problems with sensory-driven actions, which is that because of unavoidable delays in our sensory motor system, our sensory inputs tell us where things were, not where things are. So how do our brains solve this problem? This is the perfect kind of problem for our brains to solve, and it's a very important one for our brains to solve, because this is how we act in the world. It's very uncommon that we're interacting with stationary objects. So how do we get our hand not to go to where an object was, but to where it will be? And the answer is pretty simple. Instead of moving our hand to where it is, move it where it's going to be. And to do this, our brain has to be capable of predicting the sensory delay. So how out of date is the sensory information I'm seeing in this instance? The motor delay, how long will it take me to move my hand there? And the ball's trajectory. And this talk is going to focus on these first 2, the sensory and the motor delay, and really how the brain predicts these two delays. So sensory motor delays. Let me just give you the simplest example of a motor command. So in our brain, we have some motor center that has neurons that synapse onto a muscle. And if we imagine what the activity in that motor center looks like relative to muscle contraction or movement, we would expect a pattern of activity that looks something like this, which you can see with this red line. So you'd expect a burst of activity at some point in time prior to the movement. And this is indicative of, a motor command. And the amount of time before the movement that we get this burst of activity, we could call the motor delay. On the other side of things, all of our muscles have these things called muscle spindles, which are proprioceptive sensory receptors that send a long pathway through the spinal cord to some sensory area in our brain. And this is the sensory pathway, and it also is associated with a delay. So if we look at the activity in that sensory area, we'd expect to see something like this green line up here, where there's a movement, there's some delay, and then there's a burst of activity. And this would be what we would consider our sensory delay. And these two delays result in a highly predictable pattern of activity and a highly predictable type of sensory feedback. If you get a given motor command, you would expect there to be a burst of activity in a sensory area some number of milliseconds after this. And so this is a problem that, like I was saying earlier, our brains are ideally suited to solve. And anytime something's predictable, you would expect our brains to be able to perform some computation to predict that something is going to happen in the future. And they do this via a clever little shortcut that they build into the pathway, which I've shown here in this blue line. So basically, every single time a neuron sends a command to a muscle, there's a branch in that axon that goes to a different part of the brain and then sends a processed version of that motor copy to a brain area. So if you notice now, I've turned the sensory area to an integration area, because now this part of the brain is integrating the output from that prediction, that blue line, and the sensory input. And so I'm going to ask what you think the activity would look like in this integration area. But first, I want to say that this type of prediction is called a forward model. And a forward model predicts sensory feedback that will result from a given motor command and shift that activity ahead in time. So we have this forward model that's predicting sensory feedback, and then we have the actual sensory feedback getting to that integration area. And with just this circuitry, we'd expect a movement to produce a pattern of activity that looks like this, where you have some activity that kind of co-occurs with the movement because it's taking that shorter path, and then you have an echo of that in terms of the actual sensory feedback from the muscle spindle. And this is good because now we have activity that isn't out of date, that actually is anticipating the movements that are currently happening. But it's bad because now we have two different patterns of activity for a single movement. And so it's been hypothesized that our brains do one more step to really solidify this pattern of activity to just strictly be predictive. And that's to copy this forward model, delay it a little bit, and then inhibit the sensory feedback. So now we have three lines going into this integration area. The forward model, which should co-occur with the movement, and then sensory information and sensory inhibition that, if they're the same, will cancel each other out. So then we would expect the activity in this integration area to look like this, where you just get activity during the movement and you don't get, as long as expected and actual sensory feedback match, I've grayed out that second bounce because we don't expect to see any activity when the expected sensory feedback occurs. And this combination of two internal models was first hypothesized to take place in the cerebellum in 1993, this combination of a forward model and then later sensory inhibition. And in this paper, they actually also predicted that this kind of an internal model, this forward model and sensory inhibition, can't be pre-programmed. It has to develop. And this is because when you think about it, those sensory and motor delays will change across development as an animal's limbs grow, as the axons become myelinated. There's all kinds of reasons that we would expect this to be dynamic. And so there's not really a way to pre-program this model in an animal's genetics. And so when considering like what kind of movement, how could we even calibrate a system like this, they produced the following quote. The size of the feedback delay time, that sensory motor delay, could be estimated by measuring the delay between issuing a motor command and assessing its result. This would be most easy to do if the motor command were discrete. For the reafferent or sensory feedback would then change abruptly. So what they're basically saying is, in order to develop and calibrate a system like this, you'd want to produce discrete movements that generate a very brief pattern of sensory input that then can be used to estimate the delay that I was talking about. And so in the decade and a half since this paper came out, there's been lots of people that have studied internal models. So what do we know about them? Well, because they're like all neural activity, internal models of movement are noisy, researchers often need animals to do the same movement hundreds or even thousands of times to reliably determine how a given movement produces a pattern of neural activity. And to do this, it requires, this is an example from an optogenetic experiment, and it requires maybe a week of paw preference training, 2 to 3 weeks of actual training, then a surgery, and then retraining, and then the actual experiment. And all in all, this can take weeks or even a month to get an animal to reliably produce the same movement, so then you can start to explore the internal model of movement that this animal generates. And an unfortunate side effect of this is that it makes training or it makes studying the development of internal models difficult, if not impossible, because if you want to know, do you see internal models of movement in a 16-day-old mouse or rat, by the time you've trained it to produce a movement, if you can even do it with an animal that young, it's no longer a 16-day-old rat. So because of this, there hasn't been really much study in mammals about how these internal models of movement develop. So how might we go about this? Well, if you wanted to study an internal model during development, you'd need to find a self-generated movement that is produced spontaneously hundreds of times a day. That way you can get, good, reliable neural data, is discrete and highly stereotyped. That discreteness is part of that prediction from that 1993 paper I talked about earlier, and that actually drives neural activity. The first two are useless if it doesn't drive any activity in the brain. And we believe in the Blumberg lab that we've identified the perfect candidate movement for this, and that is in the form of myoclonic twitches. So here I have my other dog, Stella, in REM or active sleep, displaying this behavior, myoclonic twitches. Anyone with a pet at home, I'm sure, has seen their pet doing this when they're asleep. These are brief movements of the skeletal muscles throughout the body. Stella in this video is about 8 years old, so they obviously persist across the lifetime. And we believe that they meet all three of these criteria because they're also produced extensively in early development. And so this talk, the rest of it's going to be outlined in confirming that twitches meet these three criteria that I talked about earlier, and then finally asking the question, are they represented by internal models of movement? And if they are, we should be able to see the development of these internal models. So all of the work that I'm going to be talking about here is done in developing rats between P12 and P20. And the last talk did a great job at the development of rats and mice is pretty similar in terms of postnatal days triggering or corresponding to like certain developmental events. And so in rats, as was the case with mice, there's a lot going on here. And you can just see that in these pictures. At P12, their eyes and ears are closed. And by P20, they're moving around, whisking, doing all the behaviors that you would associate with adult mice or adult rats. And I mentioned that I'm going to be looking at the activity produced by myoclonic twitches. So I want to show you what that behavior looks like in these rats. So this is a 20-day-old rat in the mobile home cage. I have the one that has clear sides so we can get good video. And what I'm going to show you is just 6 seconds of this animal in REM sleep. And there's going to be a good deal of forelimb twitches. Over here in yellow in the corner is the time. This data was collected at 100 frames per second, but you're not going to be able to see that here. But just pay attention to how quick the forelimb is twitching. And just as a warning, I made to draw your eyes to where movement's taking place. Anytime there's a pixel that changes frame over frame, I'm going to color that red just to show you where the movements are occurring. So here is 6 seconds of twitching in a 20-day-old rat. You can see that they're pretty fast movements and that, there definitely is a lot of movements in just this 6 seconds. So I'm going to slow that down now to 1/4 speed so you can appreciate just that these are discrete individual movements of the, in this case, right and left forelimb. So here is now 1 second corresponds, or 1 second here corresponds to 1/4 of a second. And you can see forelimb twitch, forelimb twitch, forelimb twitch. These movements are pretty continuous, and there's a lot of them in just this brief period of time. So this is the behavior we're going to be looking at. And as I mentioned, all of these data were collected in the mobile home cage. The people at Neurotar were really nice and helped create a custom head plate that would work with these juvenile and pre-weanling rats. And so they were head fixed while we were doing neural recordings, and they were free to sleep and wake however as much as they wanted. And data were collected continuously for 3 to 6 hours. And these were all acute recordings. And then, like you just saw, the pup's behavior was recorded using high-speed video, and this video was synchronized to the electrophysiological data. So where was I recording? Well, if I go back to the diagram I showed you earlier, you'll see that what I want is an area that receives both sensory inputs and this cerebellar input that could transmit this internal model of movement to that area. So an area that integrates those signals. And so I chose the sensory, or VL, the ventrolateral nucleus of thalamus, because it receives both cerebellar inputs and sensory inputs. And I also recorded from VP, which is a classic thalamic nucleus that is thought to transmit somatosensory information from the periphery up to cortex as kind of a control, because it does not receive those cerebellar inputs, it only receives sensory inputs. And VP and VL are known as sensory or somatosensory and motor thalamus, respectively. So what do the data look like? Well, first, because I'm interested in behavioral state, it's important that I know that the animals are asleep when I want to, say they're asleep. And so one of the first bits of data that I collected, or one of the important bits of data that I collected, is the nuchal EMG. And this is because during REM sleep, our skeletal muscles show something called muscle atonia. If you've ever tried to fall asleep on an airplane without supporting your head, you'll be all too familiar with atonia, because as you fall asleep, all of your muscles stop being tense and just relax. And that's when your head falls and you can't fall asleep. And then you wake up and wish that you spent the extra money and gone in first class. So it's the same with rats. And this is the electrical activity of the nuchal muscle of a 12-day-old rat, where you can see that there's very little tone except for these brief bursts of activity. And those are neck muscle twitches, because like I said, they occur in all skeletal muscles. And so this indicates that, yes, this rat is asleep. On top of that, I did a movement analysis on the video, which just looks at those pixel over pixel or frame over frame changes of pixel intensity as a proxy for movement to determine if the rat is moving different parts of its body. And using that video, I scored forelimb, hindlimb, whisker, and tail twitches, which you can see here. And so there, in this chunk of video, there's lots of forelimb, a couple of hindlimb, and then whisker and tail twitches. And then finally, I recorded the activity of thalamic neurons, which you can see at the bottom here, where each one of these lines is an individual neuron in thalamus. And I did this in 12-day-old rats, in 16-day-old rats, and in 20-day-old rats. So now let's go back to this outline and confirm that twitches meet these different criteria. And first, that they're produced spontaneously hundreds of times a day. And so to convince you of this, I'll just show you each one of these blue circles corresponds to an individual animal that I recorded for several hours. And this chart reflects something about REM sleep, which is that the older rats, mice, and even humans get, the less REM sleep we produce. And this is particularly pronounced early in development. And so in these recordings, I got about 2,000 twitches, or I scored about 2,000 twitches at P12, about 1,000 at P16, and about 500 in P20. So this number is decreasing, but still at all of these ages, these recordings are long enough to get hundreds of instances of movement. And so I can definitely say that twitches meet this first criteria. So now let's take a look at whether they're discrete and highly stereotyped. And to do that, I'm going to first show you an individual twitch, this time of a 12-day-old rat pup. So here you can see its limb moves up and then goes back down. Very basic movement. And when we look at this twitch, what we can do is we can take hundreds and hundreds of instances of them and just look at the movement profile. And that will look something like this, where you can see that there's really not much movement until the onset of the twitch, and then there's this apex, this maximum displacement of the limb or whisker, and then it will return back down to a baseline. And what I can measure on this is the width at half height, just as an estimate of the duration of this movement, and see if that's consistent. So this is now one of those curves of a 12-day-old rat for forelimb movements, and you can see that the width at half height is 64 milliseconds. And if I look at that for a 16-day-old rat or a 20-day-old rat, they're all pretty consistent at about 65 milliseconds. And if I look at that for all these different parts of the body across all of my rats, this is now a violin plot. What you see is that there's not much of a developmental change. The red solid line shows the median of each group, and the gray outline is kind of the distribution of the data. And you can see that they're very consistent across age, and the dotted line shows the grand mean. And there's really not a developmental change of these movements, and they only last about 60 milliseconds using this measure. So they are definitely discrete, and they are developmentally consistent. So I'm going to say that yes, twitches are discrete, and they're highly stereotyped across the ages that we're looking at here. So now let's see if they drive neural activity. Now, previous work in the lab has shown that in 12-day-old rats, twitches do drive neural activity. So we weren't very surprised to see that they do in 12-day-old rats using the mobile home cage. And this is what the mean response to forelimb twitches of forelimb responsive neurons looks like. And this is it in VP. And then we also see in VL, we also see that there is a response of these neurons to forelimb twitches. And I mentioned that these are forelimb responsive neurons in order to show you that these neurons show what we call somatotopic precision, which is that the forelimb responsive neurons actually don't respond to twitches of other parts of the body. And to demonstrate that, I'm going to show you the same neurons' response to twitches of the hind limb, whisker, and tail. And you can see that there's very little activity here. So these neurons respond strongly to forelimb twitches, but not to twitches of other parts of the body. So in that way, it's driving somatotopically, these neurons are being driven by somatotopically restricted movements. So, and that's true at P12, at P16, and P20. But that's just one measure of precision. We can also ask whether or not the duration of the activity matches the duration of the movement. And to do that, I'm going to go back to show you the duration of these movements, which, if you remember, is about 65 milliseconds. And we can use the same measure to measure the duration of neural activity. And so in VP, what we see is that at P12, the duration of neural activity is a bit longer than the movement at about 100 milliseconds. And then it gets briefer and briefer at P16 and P20, to where the overall pattern of activity is longer at P12 and much more variable. But by P16 and P20, the duration of neural activity in VP matches the duration of the movement itself. And VL actually shows a pretty similar pattern, where you initially have a much longer response, and that response gets shorter and shorter from P12 to P16 to P20, and you see a developmental profile that looks like this. But in both areas by P20, the neural activity's duration matches the movement's duration. And so in this way, we can say definitely by P20, twitches are driving precise neural activity in both of these two thalamic nuclei. So that means that we can actually use these movements to ask the question, are twitches represented by an internal model of movement? And if they are, we can use it to study the development of these internal models of movement. So how do we determine whether this twitch represents an internal model? Well, we compare the timing of the neural activity to the timing of the movement itself. And if the neural activity precedes the movement, we can make an assumption that that might be contributing to the motor command, that it's a motor response. If the neural activity occurs at the same time as the movement, then it might represent a motor copy or a downstream computation of that motor copy, like an internal model. And then finally, if the neural activity follows or lags behind the movement, then we might expect that to reflect a sensory response. And what we can do is we can look at the peak of each neuron to determine which one of these categories it belongs to, and that'll tell us what this area is doing. So let's just look at one example. In this case, another VP neuron, or sensory neuron, or neuron from that sensory area at P12. And this is what this particular neuron looks like, with the movement in gray and the neural activity in black. And so this neuron clearly lags behind the movement in terms of its activity. So we would probably classify this one as sensory. But since we want to look at lots of neurons at once, I can color them based on the amount of neural activity and then create a heat map of all of my P12 neurons in VP. And so the dotted line shows you where the peak displacement of the movement is, and the yellow line shows the peak of each individual neuron. And if I then color these by these different categories, and so I stretch out that yellow line here, I can see that in my data, about 83% of my neurons at P12 in VP, we classify as sensory using this method. And that's good, because I told you VP should only be getting sensory input, and the vast majority of neurons are sensory. And that's true at all three ages. So at P12, P16, and P20, VP seems to show sensory responses. So what about VL? Well, at P12 and P16, VL is showing these similar pattern of activity as VP, but at P20, something changes. And then we see 50% of neurons that are too slow to be too slow to be representing like motor commands, and too fast to be sensory responses. And so this tells us that something's going on in VL and that these neurons don't really look sensory anymore at P20. And we can see the same thing by just looking at the peak activity relative to peak displacement. So instead of treating these as categorical variables, we're just appreciating the diversity. And so these are now violin plots showing when peak neural activity occurs. And you can see at P12, P16, and P20 in BP, activity lags by about 30 or 40 milliseconds. And in VL, that's true at P12 and P16, but not at P20. And that's when neural activity and movement seem to be occurring right on top of each other. But this is just one way, and we can actually ask this question a different way as well, which is, if this activity in VL is really a forward model or an internal model, then we should be able to eliminate it by blocking cerebellar inputs. And so what we did next is injected either mucimol or saline into the deep cerebellar nuclei. And those nuclei are the only output from the cerebellum. And so by putting mucimol, which blocks neural activity, into the deep cerebellar nuclei, we should be able to block the forward model of movement, but not the sensory inputs in VL. Or to put this another way, VL should look more like VP if this diagram is to be trusted. And so that's what we did. And this is what the saline injected animals look like. And the good news is that's actually very consistent with the previous data that I showed. So these data are replicating into back-to-back experiments. So the saline group just looks like the previous group, with about half the neurons looking to be corresponding to an internal model of movement. So what does the mucimol group look like? Well, after injecting mucimol, now about 59% of neurons in these animals appear to be lagging behind the movement and are classified as sensory using these criteria. And if we just look at the timing of neural activity, we see the same thing. The overall distribution of neural activity in the mucimol injected animals lags behind the movement, but not true, and that's not true for the saline injected animals. So what's going on here? Well, this is just a diagram of the summary of these results, where you see that the movement occurs at the top. The saline, and I should say that in the saline and mucimol injected animals, there was no change to the twitches themselves. The kinematics of the movement and the production of the movements were unchanged. And so you can see that the movement and the net VL activity in the saline injected animals are occurring at the same time. But in the mucimol animals, we actually saw a decrease in overall activity and also this rightward shift, or this delay of the twitch-related activity. And so what we suspect is happening is when we remove the input from the DCN, or the deep cerebellar nuclei, what we're actually doing is removing a pattern of activity that has two peaks: an early upward deflection, and that's that forward model, and then a later downward deflection that counteracts the sensory inputs that VL is getting. So in the saline animal, you get this increase and then decrease in activity. And in the mucimol animal, because you've removed all that activity, all that remains is the increase from the sensory inputs to VL. So all of this is to say, so yes, this tells us that twitches are represented by an internal model of movement. And together, all of this tells us that internal models of movement do seem to emerge in rats between P16 and P20. And that twitches can be used to reveal the development of these internal models. And then finally, the cerebellum seems to be creating both a forward model and canceling expected sensory feedback. And so it's working in that way that was hypothesized back in 1993. And I want to return to that hypothesis and give you this quote one more time, because what's remarkable about this is that this prediction was made without knowledge or without assuming that there was a movement that met these criteria. It was supposed to be purely hypothetical. But because what they're describing when they say this would be most easy to do, it would be most easy to develop a prediction of these timing delays with a motor command that was discrete. What they're really saying is that this would be the ideal movement to allow this type of system to develop. And so what we want to do in the future is to test whether twitches actually are necessary for creating and calibrating the timing of these cerebellar models of movement. So we've shown that we can use them to see these models, but what we really want to know is whether or not they are necessary for developing these models. And so with that, I'd love to thank the entire lab, especially Mark and Greta, which you can see here, who made all of this research possible. And if anyone has any questions, they're of course free to email me at my email address there. And one final thing I'd like to say is that I am on the job market right now. So if you think this research would fit in well with your department, please also feel free to email me and let me know, and I'd be glad to apply to that job. Thank you very much.
Fantastic. Well, thank you, Jimmy, for a really fantastic presentation. Just before we move on to the Q&A to address some of those questions, we do have just two last audience polls here. So we'd just like to know which statement here best describes your situation. So let us know if you currently work with awake rodents and you're interested in head fixation techniques. If you plan on working in awake rodents in the future using optical or functional ultrasound imaging, if you plan on doing so in the future but with electrophysiology, or pick that last option if you just have a general interest in the subject and technique. And like before, we're just going to keep the polls open for a few moments to collect a few more of those answers. All right, last question of the day, folks. So here, just let us know which of the following behavioral readouts you'd like to combine with imaging or electrophysiology. So here, please select all that apply. There's a number of options here. So again, just choose whichever ones are applicable to you. Pick other if there's something else that you'd be interested in, and let us know what in the ask a question box, or again, you can check that last option if none of the above. All right, fantastic. All right, big thanks to everybody for participating in the polls today. We're now going to move on to the Q&A session. And as a reminder, if you do have a question you'd like to ask, do submit it using that Ask a Question panel. And please note, if we don't have time to address your questions, we will be following up shortly after the webinar with a Q&A report to answer some of those questions we didn't have time to cover. All right, so ready to answer a few questions, Anna and Jimmy?
Yeah.
Perfect. All right, we'll try to switch off between questions for Dr. Cavaccini and then Dr. Dooley. So first one here. All right, so the striatum is composed by two main neuronal subpopulations showing different function in motor control. Do you think that the specific engagement of one subpopulation compared to another changes over development? And so, Anna, I'll direct that question to you.
Okay, yeah. In the striatum, there are two main subpopulation, neuronal subpopulation, D1 and D2, depending on the dopaminergic receptor they express on the membrane. And actually, there is a paper, there are a few papers. One that comes to my mind is a neuron paper from Kreitzer Group of 2015, where they were showing anatomically that the D1 and D2 are differentially innervated by cortical areas in adult mice. Actually, they reported that sensory cortical areas preferentially innervate the D1, while the motor cortex preferentially innervate the D2. This study was not functionally, but this might suggest that yes, depending on the time window of considering over development, there could be a different engagement of the two neuronal subpopulation. Actually, it's one of the things that we're trying to understand.
All right, fantastic, great answer. Next question, Jimmy, this one's for you. Do you think the function of twitches changes after the development of internal models? So in other words, do you think twitches continue to calibrate the system or do something else or maybe stop having a function entirely?
That is, that's the question that I'm hoping that will drive my research moving forward. One thing that we know from a lot of the research the lab's been doing is that early on, it doesn't seem like twitches are really working as a timing signal. They look like they're helping develop those somatotopic maps. And then what I was trying to show here is that they might play a function in kind of calibrating the timing of the system in that late second and then third postnatal week. And what I would love, like I was saying in that last slide, what I'd love to be able to demonstrate, because twitching persists across a lifetime, is whether they continue to calibrate that system. And that's what I'm biased to believe. But those are the kinds of questions that I'm hoping to answer moving forward.
Fantastic. All right. Great answer. Anna, this next one will be for you. What thalamic nuclei are involved in motor control?
Yeah, so in adult mice, always the parafascicular and the ventroposterior region of the thalamus show an activity related to the initiation and the execution of movements. But in my knowledge, nothing is known about the role played in motor control by those areas early on.
Excellent. All right, here's one for Jimmy. Do you see any evidence of the difference in activity between VP and VL for wake movements, or was it specific for the twitches?
That's something that I think actually highlights why we think twitches are the best movements. So that's something that I looked at. But the problem is, because these are young animals and you can't really train them to do a wake movement repeatedly, there's so much variability. So I can trigger on the initiation of wake movements, but there's so much variability in what those wake movements are. Sometimes they're grooming, sometimes they're locomotion. And so the timing of the movement and the different muscles that are being used in that movement are so variable that you can't really get the resolution necessary to see, a 20 or 30 millisecond difference in activity. So twitches are the best movement to be able to see that because they, only affect one muscle at a time. And they're so brief that you can really see, with this movement, is there activity afterwards? And with wake movements, it's just not possible to see a difference. There might very well be one, but I don't think using that method, we'd be able to detect it.
All right, great answer. All right, Anna, next question here for you. Have you tried to stimulate a cortical sensory area to check the response in striatum early on?
Yeah, so I tried to evoke sensory response in striatum early on, actually through whisker stimulation in awake fixed mice. And I stimulate the whisker by sending one stimulus every 20 seconds for 20 times. And then I record the multi-unit activity. And at the 3-time windows that I have presented in the talk. And what I have seen is that I was able to evoke a response at postnatal day 17-24, but not before. So it looks like sensory input, at least coming from whisker, reached the striatum after the appearance of the whisker-dependent sensory behavior. So at least considering those inputs, striatal activity doesn't seem to be engaged by them early on.
Fantastic, great answer. And I think in the interest of time, we'll just have one last question here. We'll end on a great one here for Jimmy. So this goes back to the slides you showed at the start with the toddler and Zelda. So as opposed to the twitches, they both use their visual system. So how do you think using vision applies to these internal models?
That's something that I'd probably have to speculate more on, but what I would guess would be the case is that, these internal models and these representations of the world and kind of predictions of sensory feedback, they start very simple and then they can get more and more complicated as animals or people get more experience in particular situations. And so my focus here on the sensory motor system and kind of predicting these sensory motor delays, I think that is the base model that then you can add additional predictions from visual input, from auditory input, once you have that base level set up. And so while I started showing, two different, my dog and a toddler doing these visual tasks, I think prior to even that, they have to set up this basic internal model of, predicting sensory motor feedback or more sensory feedback from a motor command. So I think that's the way. That's the way that I think about it. And I would guess that once that's set up, the same kinds of things can be used to then predict visual input or auditory input or any other modality.
Fantastic. Well, thanks so much, Jimmy, and thanks so much, Anna, for the really fantastic presentations. It's been a real pleasure having you with us.
Yeah, thank you for having me.
Thank you.
Yeah, and a big thanks to everybody also for joining us today to attend the webinar, and we look forward to having you with us again soon. Have a great day, everyone.